Hello, I'm Tristan Cai from the AAVAB Laboratory. Our company is based in Shanghai, China. I'm very glad to share our work about the leading-edge platform we built up for the AAV capsid engineering. Deliver the healthy genes into patients is the basic idea of gene therapy. Very straightforward. AAV is one of the vectors to transfer the genes into patients with several advantages, such as good transfection efficacy, and it is not related to non-human disease. And in nature, lots of AAV exists, which provide a large pool for proper vector selection. And the genome of AAV can form episomal for long-term expression of the therapeutic genes. And the immunogenicity of AAV is relatively low, comparing to other vectors such as lipid nanoparticles or the adenovirus. Of course, it is not non-immunogenic because it is still an exogenous protein. Till now, there are several AAV-based drugs have been approved in Europe, in USA, and China. The AAV-based gene therapy really benefits the patients who have no or very limited therapeutic drugs. However, off-targeting is one of the key issues for AAV vectors. The commonly used AAV vectors can infect multiple tissues in human, which is caused the multiple tissue tropism. Because of off-targeting, the AAV particles which goes into the red tissue were reduced, and higher dosed AAVs are necessary for sufficient gene delivery, which results into the higher cost of AAV production and raises a safety issue. In addition, the expression cassette requires complicated design to increase the sufficient gene expression because of the loss of targeting AAVs. So the AAVs which can specifically bind to targeting tissues is required. Many efforts have been done to enhance the targeting efficacy of AAVs. As we all know, the capsid of AAV mediates the targeting ability by binding with the membrane receptors. The genome of AAVs contains one VP gene, which codes for three VP proteins, namely VP1, 2, 3. In conventional capsid engineering method, the AAV capsid library are generated by aeroprone PCR, shuffling or peptide display, and then screening by model animals. By several rounds of panning, the specifically enriched capsid in target tissue were harvested by RNA sequencing. However, there are still some drawbacks. First, the screening is done in animal, which is quite different with human. The transfer rate for what we find in animal to human is very low. Second, all three VPs are changed simultaneously, which limited the range of modifications in VPs. If the new VPs contains many mutated sites, the packaging efficacy will be dramatically reduced. Further, it is not possible to introduce different modifications in different VPs to get the chimeric AAV particles, which we will discuss later. Focusing on these problems, we develop our AAV engineering platform. First, we use a human pluripotent stem cell-derived tissue organoid for library panning to avoid the interspecific difference. Second, we build up the versatile AAV engineering platform via the separate capsid protein expression, which give a high freedom for VP engineering. Here is an example for human cardiac-specific affinity peptide screening. First, the HPSAs were differentiated into human hepatocyte and cardiac organoid. The cardiac organoid are multiple-chambered and contains a cardiomyocyte and blood vessels, which is structurally similar with human heart. Then, the negative selection of peptide library by human hepatocyte and the positive selection by human cardiac organoid were used for cardiac-specific affinity peptide screening. After 4 rounds of panning, the peptide which with high catalytic specific index was screened out, together with the ones specific for human hepatocyte. Now we are doing the validations. Here is our versatile AAV engineering platform with separated capsid protein expression. As shown previously, AAV particles contain 3 VPs, which are derived from the same gene. Different VPs have different structure and functions. For example, VP 1 and 2 are mainly mediate the endosome escape, while VP 3 is a scaffold of the viral particles. For small peptide insertion or modified the VPs with not so much regions, the packaging efficacy is okay. But for large protein insertion, such as nanobody or the SAFV, or modified the VPs at several regions, the packaging efficacy is significantly reduced. To increase the tolerance to the modifications in VPs, we developed the separate VP packaging platform in which VPs are expressed independently. Thus, we can only modify the desired VP, while leaving the remaining VPs unchanged or less changed to keep the packaging efficacy. By plenty of mutations and combinations, we finally find the best plasmid combos for AAV6, AAV2, AAV5, AAV9, with VP isolated and AAV packaging unaffected. Here is the isolated platform for AAV6. As shown in Western blot, the VPs are separated quite well for the packaging test. Not all combinations with all 3 VPs are maintained, such as this combo. The packaging of AAV are severely affected. This might be caused by the improper mutation, which may affect the other key AAV protein expression. The separation platform is also built up in AAV2, AAV5, and AAV9. To test the versatility of this platform, we insert an VHH at all of the loops in the VP2 of AAV6. If we insert a VHH in all VPs, the viral particle cannot be packaged. In our platform, the VP2 contains a VHH, and it can be incorporated into the AAV particle. By the surface displayed VHH, the infection of AAV is significantly enhanced to about 3-fold or 5-fold. In summary, we generated the human tissue organoid-based screening platform to get the AAV vectors, which is real infection to human cells, and the capsid engineering platform with advantages such as it allows a modification of VP protein independently to get the AAVs with large protein insertion or with chimeric VPs for different functions. And the enhanced targeting efficacy were verified by displaying the weightage on the surface of AAV6. We are developing the novel AAV vectors such as T-cell targeting AAVs which can cross the brain-blood barrier and other T-cell targeting AAVs. Finally, as our goals for better gene delivery, we believe that our platform can promote the development for AAV drugs. Thank you for listening, and we are open for cooperation. Please contact me with the following email for more information. Thank you.